The next talk we have is from a member of our scientific advisory board, Dr. Rick Levy, who is a child neurologist and neurogeneticist and neuroscientist at Stanford University at Stanford Medicine Children's Health. So she'll be talking to us about CACNA1C related disorders, the genetics of that, and how we could potentially translate that to things down the road. So thanks, Becky.
Thanks so much, Jack, and thank you, Sophie, and I want to thank all of you for being here. This is such a pleasure. In the interest of time, I might jump over or kind of limit what I say about a few things here, but I'm always happy to go back to it in our questions. So as Jack mentioned as kind introduction, I'm a neurogeneticist in California, in the United States, and I'm going to wear a couple of different hats as I talk about some things today. I work in Dr. Sergio Pasca's lab out here at Stanford. And so this is how I envision translation as a path from where we go from the bedside, meeting an individual and their family, over to the bench where we do science, and then back again. And so especially in rare disorders such as CRD, we have a lot of gaps in our knowledge, and you're hearing about many of those throughout the day today in this conference. Sometimes it's about phenotyping or understanding the symptoms of individuals with the disorder. Sometimes it's about how do we make that diagnosis? How do we understand the variant of uncertain significance? How do we make model systems, and how do we apply them to understand the pathophysiology, which is our fancy way of saying what is it that causes a disorder? And once we know what causes it, then we can identify potential treatments that we'll try to bring back to families. And so today I'm going to try to wear both of my clinical hat and my scientific hat, and we're going to talk a little bit about how do we understand a genetic test report, as well as how can we translate scientific findings into clinical changes. And very briefly, there's some spectacular speakers later today who will go into this a little bit more, but the gene CACNA1C encodes what we call in science the CAV 1.2 or L-type voltage-gated calcium channel. And what this does is, hopefully you can see my pointer, it takes calcium, one of the salts in our body, and allows it to come into cells. And this is really crucial when we think about excitability in the heart and in the brain, as well as many of our other organs. And these types of calcium channels help these cells to potentiate or to increase their firing in response to signals. So you're seeing here this potentiation that's happening. Those spikes are getting faster and faster. And likely as a result of tissue-specific expression, we can now recognize that CRD is a spectrum of cardiac symptoms, neurologic symptoms, syndromic symptoms, and everything in between. And I'll jump over this because you folks are very familiar, and I'm really thrilled. I wasn't sure whether or not the new consensus image of how we're imagining CRD was ready, and I was so glad to see that that Jack was able to share that with us this morning. So quickly, I'm going to take us back for a moment and talk about some of the genetics terminology or words that we use that might be on the results that you're getting for your child or loved ones. And when we think about our genetic information, we all have two alleles or two copies of our chromosomes, our DNA, our genetic information on the ones that are what we call our autosomes, so not our sex chromosomes. And nowadays we use the term variant. In the past, people would have said mutation, but we try to use the more kind of neutral term variant. We specify if that variant is benign, if it's unknown, or if it's pathogenic, meaning disease causing. And I'll come to that in a moment. And heterozygous means when only one allele or one copy of our gene has a variant in it. Homozygous is the term when it means that both copies have the exact same variant. And then there's something called compound heterozygous, which means that that there are two different variants, but in the same gene. Those can either be on your two different copies, which is called in trans, or they can be on the same copy, which is called in cis. And this means that someone inherited one from each parent, or that they had a new change that happened one on each copy, whereas these were inherited together. And so parental testing is probably something that your doctors might have talked about because it helps us to understand how some of the differences about how these variants can be inherited over time. So a variant just means a difference from the reference genome, and we each have millions of variants. So how do we know which ones affect our health? And this is an example of kind of some of the scoring schemas that are agreed upon criteria for how we rank a variant from benign, which means we're pretty sure it has no effect on someone's health, up to all the way up to pathogenic, which means we are pretty certain that it impacts someone's health. And as many of you may have experienced, a variant of unknown significance is what makes everyone groan, both your healthcare providers and maybe even yourselves, because it means that sometimes we're not entirely sure what that impact might be on our health. There are a lot of ways that I can get into if anyone has questions about how to think about if a VUS or variant of uncertain significance is important, including testing other members of the family, looking in larger databases, and using a variety of model systems and predictions to test if there's a functional impact. And you might have heard the term inheritance or dominant disorders when people talk about CRD and Timothy syndrome. And what that means is that here's each one of us, here's a couple here, and dominant means that one variant is enough to cause someone to have symptoms of a disorder. This can mean that there's a 50% risk of transmitting that variant to children in every pregnancy. Very briefly, if you've ever heard the term recessive disorder, that is where someone needs to inherit two different alleles, or two different variants, excuse me, one from each parent, and only then will someone have the symptoms of this disorder. And CRD is a dominant disorder, so over here, meaning that just one genetic variant is sufficient to cause someone to have symptoms. But in many ways, we're still understanding why different variants can affect just one part of the body more than another, or one person more than another. And we know that that has to do with a lot of functional impacts of what these variants do. So this is an image I was able to find. Here's a sodium channel, here's a potassium channel, but just really like the way this kind of walked through, even though we're talking about a calcium channel today. This is an image of what these might do to the heart. And some variants can cause what we call a gain of function. So here is a channel, here's typically a little bit flows through. With a gain of function, more is going to flow through, and that's going to impact the electricity in those cells. So in this case, the channel is just open. There can also be loss of function, in which case less is flowing through a channel, and it also impact electrical conductance. But it gets sticky because sometimes there's in this case, there's a little gate, which is very similar to what happens with CAV 1.2. And what they're saying here is that actually this gate is broken. There's delayed inactivation, and that actually leads to a gain of the channel function, but a loss of function in how the channel gate is working. So there can be a lot of confusing terminology that makes it hard sometimes for us to understand what exactly the net impact is on the cells in our body. So these terms gain and loss of function can overall be very complex. But if we're able to understand, either from experiments done in cells or from someone's health information, or we're trying to work on things like predictions. There are a lot of people who study how to model genetic variants and protein variants to predict what would happen to function, that if we have access to that information, it might help us determine what could be the most helpful way to treat someone's symptoms, because we'll know whether we're going after a gain or a loss of overall channel function. Very briefly, with many of your help, those of you who participated, and as well as the support of the TSA and the TSF, we've been trying to understand a little bit more about the neuropsychiatric symptoms over time that people are experiencing, and whether or not this is really tied just to the cardiac symptoms or whether there's kind of independent factors. And we know that overall that these symptoms are relatively common. This does not mean that everyone with CRD has neurologic, developmental, or psychiatric symptoms, but it just means that, that it's important that we look for them, and that it's not only related to whether or not someone has had underlying cardiac issues and has had, for instance, injury from cardiac arrhythmias in the past. And when we establish this baseline of what neuropsychiatric symptoms might be common that we might want to look for, this can give us clues into what brain regions are affected, what diagnostic clues we could use to help get to that diagnosis earlier, what treatment approaches we should consider, and what symptoms we might want to track over time in order to determine the efficacy of a treatment. So given the high rates of neurodevelopmental disorders in CRD, it's really important that we be able to investigate the mechanisms of how calcium signaling affects neurodevelopmental disease and potential therapies. And by understanding this, we can think about how we try to treat those symptoms. And then we could determine what people in medicine often talk about a therapeutic window, which means if I intervene, if I give a treatment or a medication right now, is it going to be able to change what happens down the road in that person's health? Can it significantly improve the symptoms that they're having? So next, I'm going to explain very briefly what organoids are and how we've been using them in the Pascha lab to model the Timothy syndrome type 1 variant in particular, as well as other aspects of CRD, and how we're trying to understand the neural pathophysiology. And so very briefly, if this movie plays, oh good, it's playing. 3D neural organ model systems is a platform where we take stem cells, we take skin, we revert them into stem cells, and then we direct their differentiation towards neural cells. And then actually, if instead of letting them grow flat, we encourage them like this, they will actually start growing into little 3D structures or organs that we call organoids. In other words, they're like organs of our body. And these are ways for us to understand how cells from the brain or neurons, how do they connect together? How do they wire together? We can use these to think about different parts of the brains. So here is one that has been patterned to look kind of like the cortex or the outer part of our brain. We can also combine them together into what we call assembloids or assembled organoids, where we can study how different regions of our that mimic different parts of the brain come together. And finally, we can actually transplant these into the brains of newborn rodents, and they'll actually integrate, and then we can study how they interact with information that's coming from outside and how they interact with behavior that the animals do. So these model systems can be a very powerful tool to look at how human cells impact development in the brain. So TS1, as one of the first identified pathogenic variants in this gene, has been a focus of Sergio Pascha in the lab for many years. And if we make these organoids from people without TS1 and from people with TS1, and then we try to compare them, one thing that we see here, TS1 is in red, is that gain of function, so that there's extra calcium that we're seeing inside of cells. And we can block that with things like nimodipine, which is one of the calcium channel antagonists that some of you might recognize. So we know that it has to do with the channel function. But the issue is that nimodipine and many of the other drugs that are really helpful for the heart, for those with a gain of function in this channel, they don't cross over into the brain. And so we have to think about different ways to try to help some of the symptoms in the brain. And in the human brain and in assembloid models, we can look at these. These are inhibitory interneurons, and they actually do this jumping migration to get into the cortical side of these assembloids. And this is very analogous to what happens with cells. And these types of interneurons are important for the excitation and inhibition balance in the brain. And so TS1 derived interneurons from the same organoid system. So here's control, and on the bottom is TS1. They're doing this jumping movement, and you can see that the TS1 derived interneurons are not moving as far or as fast. And we can quantify this movement, and here the TS1 is in this teal color, the control is in gray. You can see that they've gone less of a distance over these 8 hours of being watched. And this is in fact due to the fact that they jump more often, but each one of those jumps is shorter. So this suggests that inhibitory interneurons might not be integrating into circuits perhaps as well, at least in our in vitro studies. And this type of impact on interneuron integration is thought to underlie the risk of things like epilepsy and autism spectrum disorders in general, not just in CRD and TS1. And so with this model system, we're able to detect some differences in how the organoids derived from individuals with TS1 can vary in their cellular composition, their organization, and their development. And so given how advanced these model systems are, we asked, could we use these models to look for potential treatments for TS, for CRD, and then measure their effects on these same phenotypes? And in particular, given that TS1 is a gain of function variant, could we target and reduce its expression? So very briefly, we talked about DNA or genetic information earlier. And in the DNA of our genes, there are many different functional sequences. And some of those are called exons, which are the part that biologists usually talk about. that code for what becomes the protein. And introns are the spaces in between them. They were previously thought to just be kind of space holders, and they get deleted out in what we call splicing in order to make the RNA that eventually becomes the protein in our cells. But that splicing doesn't need to include every exon. And in fact, there can be alternations in which of these exons get involved, which we call alternate splicing. And this splicing regulation is something that is science in general is just kind of starting to understand and learn how to manipulate. And many genes generate diversity by this type of swapping of exons. And for CACNA1C, there are many different isoforms, many different swaps. But the one I'll talk about right now is between exon 8 and exon 8A. So you can either have one or the other, but you can't have both. And it turns out that the TS1 variant is right at the end of exon 8A in particular. And not only does it lead to the channel being open more, but it actually also leads to splicing changes such that exon 8A is actually kept more often than it should be by chance. And so here the Timothy syndrome-derived organoids are in teal, and the amount of 8A right here is increased over time compared to the wild-type derived organoids, whereas the amount of exon 8 stays mostly the same or slightly decreased. So we know that expression has differences across development. And if TS1 is a splicing disorder, then we thought about modifiers, which are known as antisense oligonucleotides. And they have three big impacts. They can actually just degrade all of that RNA. They can stop it from being turned into protein, or they can change that splicing. And so we wondered if we could develop an antisense that would change that splicing of exon 8A in order to think about reducing the expression of the TS1 variant. And Xiaoyu Chen and many others in the lab and I worked on designing antisense kind of along the area, over the area of 8A. And then we tried to figure out which ones would reduce 8A expression. And I just want to point out for a moment that this means that this particular antisense design is really focused on 8A and the TS1 variant and anything else that's in 8A, but it also represents a strategy that could be applied to other parts of the gene as well. So we found several antisense that lead to a reduction in that 8A expression while keeping 8 present as well. And when we give this antisense, which is now the purple lines, so here's the teal, which is the untreated TS1 organoids. And when we give the antisense, the calcium signal improves closer to where the wild type are. And this is also true when we look at the channel inactivation. Here is the untreated, and then here is the treated organoids with this antisense. So the channel activation rate is improving with this antisense. And then when we look at that interneuron migration assay, when we give the antisense here in purple, they start to improve as far as their number of jumps and the length of each one of those jumps. And then when we take these TS1 organoids, if we transplant them into the cortex of rat so that they can develop and mature, and then if we give them one dose of the antisense and then look about 14 days later, we can look at the calcium activity. And here we see the antisense is in purple, the untreated is in green, very elevated calcium signal, and the antisense starts to improve that calcium signal. And we can see that here. So this demonstrates that the channel function can actually be improved in the living organism. And then when we think about these neurons, these brain cells, they're really big and complex. Here is a typical neuron that's living inside of one of these organoids. Here's one of the neurons that is living inside of the TS1 organoid that's been transplanted. But after giving one injection with the antisense, this arbor, these projections, the way that these neurons contact each other is starting to increase. And so we're hopeful that this means, and this is a picture of that complexity is increasing over time in purple. And so we hypothesize that this improvement that we can see, this outgrowth, might mean that there's still an open window to be able to treat and intervene in neurodevelopment. I'm going to skip over this for the sake of time, but we're looking to see if we can find antisense that are very specific to the TS1 variant in particular. And then just to finish up, the goal, anyone who's driven by science to try to help individuals with disorders is to figure out how can we bring this back to the clinic. And theoretically, there's a lot of steps there. First, determining toxicity, pharmacokinetics, which means how does a drug or a treatment enter the body, how long does it stay there and have an impact. Then we have to think about how do we understand whether or not it's impacting someone's health, and then how do we find safety and efficacy in clinical trials. So to briefly describe a project that we've just been starting up in the lab, we want to see if we can take this antisense and understand its safety profile and whether or not it might be helpful for potentially giving to individuals with TS1 or exon 8A variants. And the first part of this will be to understand the safety. We're giving this to wild type or control mice and primates across a dosing range, making sure that there's no issues with their blood lab values, their behavior, their hearts, a variety of other types of monitoring of their brain. Next, we're trying to figure out what are the pharmacokinetics. So if I give one dose, how long does it last in their bodies? How long is it present in their brain? How much does it penetrate into the brain? And then finally, we're trying to figure out when we give this to wild type animals, how long does it impact the expression of exon 8A versus 8? What about the overall protein of CAV1.2? And we're trying to make an antibody against the antisense to see how deep into the tissue is this going. And future directions, if this proves to be safe and efficacious, would be potentially even planning for a clinical trial. And so our plan is to bridge this scientific work into a safe and thoughtful treatment approach. So first, I would want to better understand your needs, what risks or worries or concerns you would have to think about a possible neurodevelopmental treatment. I'm really honored to be able to participate with the TSA as well as the TSF, and I hope to continue these conversations about what we call a needs assessment and clinical trial readiness. As Mia St. John pointed out really elegantly in her video this morning, that also means things like biomarkers. So what are some things we can use to understand how someone is doing now, for instance, with their speech or language, and whether or not that changes after an intervention? We've been having a lot of meetings here on the Stanford campus with some of these groups to try to figure out what would it look like to even think about putting together a rare disease treatment trial. And then we've already started some conversations with the FDA to understand what type of safe profile and efficacy profile would we need to have to try to move forward with this. So in summary, I'm really thrilled to be able to try to bridge between the basic and the translational research in order to figure out how we can take things in science and really make them impact each other's lives. So first I want to thank Sophie, Jack, everyone in the TSA, Catherine and Rosie and everyone with the TSF and all of you for this opportunity to speak today and for really motivating our research because as a physician scientist, this is what makes us do things like get up at 5 in the morning and go to the lab so that then we can be here for these conferences to be able to see how our work is directly helping families like yours. I want to thank everyone in the Pashka lab, especially Xiao Yu and a bunch of Jill, Noah, Laura, and Mayori, Grant, who works with us on this clinical translation project, and Dr. Hall as well. So with that, I'll pause there.